Hi, I am Mrs. Sloan, and this is a video for my honors biology class. This is our Chapter 13 Lymphatic System and Immunity, and this is video 2. In video 1, we discussed our innate immunity, and now in video 2, we are going to discuss our adaptive or acquired immunity. So I'm going to make myself a little bit smaller, and let's see, put that away, and I will present to you. All right, so here in part 2, we're going to learn if something invades our body and we've got our innate immunity fighting, how do we make sure we eliminate it? Okay, so we're going to be talking about our adaptive or acquired immunity. And our immune system is adapting. We are acquiring this immunity by what we are exposed to. So let's review a little bit about who the players are. So we've got our general policemen. And remember, we learned all about these guys. Dendritic cells eat, macrophages eat, and they're also antigen presenting cells. Okay, here are granulocytes with our neutrophils being most numerous. Complement proteins, remember how they can drill holes. Natural killer cells kill, right? Mast cells release histamine. This is part of our general fighting force. Now, if this is all we had, for instance, neutrophils, they don't learn, they don't get better, they don't respond any faster. And so what can end up happening is, if you've ever had a sore throat, your neutrophils, they'll just say, oh, danger, danger, stranger danger. They'll start throwing bombs, and they take out a lot of our own body cells. A lot of friendly fire goes down, and they destroy parts of our throat while trying to protect us. Okay, they're just saying, oh, there's something foreign there, I need to get rid of it. Okay, and so a sore throat is an example of when your body is using its innate immune response in order to fight something off. We really need our special forces to come in, swoop in, and save the day, because they get better at it, and they will only recognize one type of pathogen, and they will fight in two different ways. B cells will ultimately make antibodies, which will tag and flag any free floating thing in your blood or tissue fluid, like you are an invader. It tags them and marks them for destruction. And then T cells, they do cell-to-cell combat, and they wipe out our own body cells that have been infected by that invader. All right, so that's the overview. They are, if we go in our notes, if you're new to my class or videos, down in the descriptor of this videos, you will see the group shared notes that my students use. Column 1 is the scaffolding. I'll help you fill that in. Column 2 is blank. I encourage you to throw in pictures that relate to what you're writing in column 1. So we are under acquired immunity, And so remember back to those antigens I talked to you about in video 1. This immunity is triggered by antigens, and that is defined as, here's your first notes, any foreign substance that triggers an immune response. Any foreign substance that triggers an immune response. And they are found on the surfaces of viruses, bacterial cells, or eukaryotic cells. Remember, bacterium is a prokaryotic cell, right? So found on the surfaces. And they are made of carbohydrates or proteins. So they're surface markers. Okay, now there are two basic Navy SEALs, special forces, okay? And they are either part of the antibody mediated team or the cell-mediated team. So I'm going to walk you through those. So antibodies is all about the B cells, and cell-mediated is all about the T cells. Now, why are they called B and T? Do you remember from video 1? B cells mature in the bone marrow, right? T cells, they're born in the bone marrow, but they mature in the thymus gland. Who are their targets? Well, the targets for B cells are any free-floating viruses or bacteria moving around in your body. Okay, the target for T cells are our own body cells. See how they're infected with the virus inside? That's who the T cells are working with. What's the process? Well, B cells ultimately will get antibodies made, and these antibodies are specific only for this bacterium, and you would have a different antibody that is specific to this virus. Very, very specific, okay? And so they will mark these guys for destruction. Whereas T cells, they're all about cell-to-cell combat and killing off our own body cells that have gone rogue. Either they're cancerous and they need to be killed, or they've been infected by a foreign object. All right. So on your notes, you can see we're going to start with antibody mediated, this whole pathway. All right. So I'm going to tell you a little story. Here we go. Here's a dendritic cell. He's part of your innate immune response, and he eats anything that's foreign. He is eating. Now, he rips it apart, but he saves the antigen so he can tell others, hey, and it puts it right on the surface. He says, hey, I have eaten this thing. I've overcome it. And so he displays it inappropriately in this picture, and I'm sorry, but it's a really good cartoon. So this phagocyte, this dendritic cell, he is going to display this antigen, and he's going to look for that one or two T cell that can, there's one or two of them that can recognize that particular antigen that we're looking at right here. I need a pointer, just a second. This particular red gumdrop antigen. This helper T cell is what's called, if it's never been exposed to this antigen before, it's called a virgin T cell, and it has been waiting your whole life, right? You have virgin T cells and B cells in your body right now that have never been exposed to an antigen, but are ready for wherever you may travel, right? And so this helper T cell only recognizes this particular antigen, and now he is activated. The light is green. All right. Now, let's talk. Here is the virgin B cell. Look at 3 different pathogens here, but he only can recognize this one. He doesn't know, he doesn't have the receptors, the B cell receptors to recognize these 2. This one fits. He has to be in cahoots with the activated T cells. They have to have agreement before you develop a whole clone army of B and T cells to fight off this invader. They have to meet and agree, yes, this is a bad guy and we have met the same one. Where might this go down? In a lymph node. Remember lymph nodes? Yes, where your tissue fluid is evaluated. And so they are in agreement. So what happens next? Well, that B cell that was activated, he will do what's called clonal selection. He'll make clones of himself, do a bunch of mitosis, and he will make 2 types of cells. He will make plasma cells and he will make memory cells. So let me talk about the plasma cells. The job of the plasma cells is to make a bunch of antibodies to go out into the plasma. Remember plasma, the fluid portion of blood, mostly water, but it has things dissolved in it. It releases antibodies into the plasma. These antibodies are specific for this particular antigen and will mark them for destruction. This takes care of any of the free floaters that are out in your body, all right? And then here, okay, these antibodies have tagged this guy, and it makes it even more yummy, even more desirable for this macrophage because he wants to eat it now. Because yes, he knows it's foreign, but it's tagged with an antibody. We need to eat this thing right here. Okay. Now the memory cells, let me go back. Okay. Remember, it makes plasma cells, a bunch of those, millions of them, literally raises a clone army of plasma cells that can make those antibodies. It also makes memory cells. Memory cells are for the next time. This is the acquired part of your immunity, the adaptive part, because after this battle is over, you can't keep 1 million plasma cells for every infection you've ever had in your blood. You don't have enough room. That would be like leukemia. So they all have to commit apoptosis, except for the memory cells. And the memory cells are not virgins. They are more easily triggered. And what will happen is they will, when they come in contact with that foreign invader again, they are, boom, the light is green, clonal selection, making B cells, making more memory cells, so you can fight the infection, and this time you'll have even more antibodies than you had before, okay? So antibody-mediated response, it targets the free-floating, these are in your notes, targets the free-floating viruses, bacteria, or toxins, free-floating viruses, bacteria, or toxins. Remember, B cells originate and mature in the bone marrow. That's what you need. When a virgin cell is triggered by an antigen, 2 cells are formed. We talked about those 2 cells, right? Let me go back here. 2 cells are formed when a virgin is triggered. Plasma cells, and that's going to produce antibodies. That's what you need in your notes, antibodies. that are specific to that antigen. When the fighting is over, what will they commit? Apoptosis in order to maintain homeostasis. All right. And then also memory cells, and the memory cells will recognize the same antigen in the future. It's for the future. Okay. So clonal selection is when you make 1 million clones of these plasma cells right here. Clonal selection are clones of the cells that recognize and produce antibodies to build up an army of cells, that do the same. All right. Now, let me go here. So this shows you what those antibodies are that those plasma cells are making. Now, there's two heavy chains and 2 light chains, and then they are binding right here. This is specific to this particular antigen. Now, the base of the chain right here, that there are like 5 different bases you could have, and this determines what happens to the invader, what it's going to trigger. Is it going to get eaten, etc.? So on your notes, antibodies are Y-shaped. Okay, they bind to and mark antigens for destruction. The mode of destruction is determined by the base of the Y. All right. So here, these are several different antibodies. One of these is part of your additional objectives. So immunoglobulin G is most common. Okay, IgG. IgM, you don't need to memorize these, but these help when in the blood, when you have a pathogen, it kind of groups them all together. IgA is in your throat. D is in immature B cells in readiness. It tells you when you're done. And this one, look at IgE. Here's an antibody, and it's embedded in a what? A mast cell. So if this antibody is connected to an antigen and in a mast cell, remember what that mast cells will release? Histamine, right? And that causes vasodilation, okay? So IgE found in blood, okay? Here it activates the complement and clumping. IgA is in your throat, and then IgE is found on, remember, eosinophils. Those are one of the granulocytes and mast cells, and this is what can cause an allergic reaction, all right? Now, here you can see some clumping. The cool thing about antibodies, because they have 2 arms, they can hang on to 2 different antigens at a time, and this kind of helps group all the bad things together, these foreign invaders that you're trying to get rid of. Here, you can see the macrophages have receptors for the base of the Y that lets them know, hey, I got to eat this thing. It is a bad guy. All right, here, connecting it to the complement system that we talked about earlier, that antibodies can work with the complement proteins in order to get those holes drilled into them. Remember, we talked about the complement proteins were part of your innate immune response. Okay, and here is IgE in a mast cell, and so you can see the antibody bound to the antigen, and this makes the mast cell release histamine, which makes it easier for cells to come to that site of that infection. All right, so that's one of your special forces. Now let's talk about your other special forces, and these are your T lymphocytes, cell-to-cell combat, cell-to-cell combat. All right, now, whereas the B cells produce 2 kinds of cells, T cells produce 3 kinds of cells, okay? Now, the virgin T cell, so here is a macrophage, okay? He's eating a bad guy. Remember, the macrophage is part of your innate immune response. And what it's doing is it's ripping it apart and it's displaying it. Now, macrophages have something called an MHC2, okay, like 2 arms to it, and it displays the antigen, and it's looking, remember how we talked about antigen presenting cells? It's looking for a virgin that recognizes this antigen. It's like it's cut off the head and said, look at these bad guys, which one of you recognizes it? When it does that, it will stimulate that T cell, and then what will happen is we'll start to do mitosis, and it'll make some cytotoxic T cells, that's right here. They're all about killing, but they will only kill cells that are displaying that bad antigen, meaning they are infected. And when they're infected, they don't display on an MHC2, they display on an MHC1. And I always remember MHC1, you're done. MHC2 is like a macrophage. I will help you. I'm going to show you more on that in just a minute. I'm just introducing it. Okay. And also helper T cells, these are like the wedding coordinator or the general, and they amp everybody up to get this immune system going. And then memory cells, remember, that's for next time. So cell-mediated immune response, it targets our own infected cells displaying a foreign antigen, our own infected cells displaying a foreign antigen. T cells originate in the bone marrow, just like B cells, but they mature in the thymus gland, When a virgin T cell is triggered by an antigen-presenting cell, like a macrophage or dendritic cell, displaying it on their MHC2, I will help you, it will mature and form 3 types of cells. Cytotoxic T cells that kill any cell displaying that foreign antigen on an MHC1. Helper T cells are like an army general, and they are encouraging / guiding the response. And memory T cells are for the next time. Okay, and remember, they're going to make a clone army of cytotoxic T cells and helper T cells and some memory cells. All right, so let's talk about T cells. A T cell could have the virus or the bacteria literally crawling on them, and they won't recognize it. Okay, the only way you can talk to a T cell is by displaying a piece of that foreign invader on either an MHC2 or an MHC1. Now, MHC1s, this would be like on most of your body cells. And so if you're displaying an MHC1, that means like, yeah, I know, I'm infected. You should kill me. I know, I'm infected. But if you display on an MHC2, you are a part of your immune system. You're part that has fought it and destroyed it. And you're saying, look what I am, look what I killed. Help me. That's when it displays. So this could be like a dendritic cell. This could be a B cell. This could be a macrophage. All right. So MHC1, okay, you're done. MHC2, okay, I will help you. So let me show you another picture of that. Okay. So let me move myself down here a little bit. Okay, here's the referee deciding if you're safe or you're out. So look here at the top. The very tops of both of these pictures are T cells, okay? So the purple thing sticking out here is a T cell receptor, okay? The antigen is the red triangle. This antigen is getting displayed on an MHC1, which is an infected cell. It's getting stabilized from the T cell by a protein called CD8. You don't need to memorize that, all right? But it will get 8. This infected cell, okay, displaying it tells a cytotoxic T cell, see how it has a little C there? A cytotoxic T cell to kill it, okay? So MHC1 plus antigen. In this scenario, this could be, over here on the right, it could be a dendritic cell, and it's displaying the pathogen on a portion of it, the antigen, the surface marker, on an MHC2, okay? And it is saying, I killed it. It's not infected me. I have, in fact, killed it, and I'm showing it to you. Can you help me? And so this is a helper T cell that it's showing to, okay? The purple, again, is the receptor of the T cell. And then this will facilitate and amp up and trigger your immune response. So MHC1, here on the left, is on almost all cells. And that means if you display on an MHC1, you're done. You can be killed if you're displaying that foreign antigen. But if you're displaying on an MHC2, that's on an antigen-presenting cell. And these would be things like macrophages and B cells and dendritic cells. All right? Now, here we go. So here's the deal. Let's look at these pictures and see if we understand it. T and B cells ignore the cell. Why? Well, look at its MHC marker. It's empty, so it's not displaying anything. So there's not a problem, and it's not trying to initiate an immune response. Okay, let's look at this one. T cells will start an immune response. This is a processed antigen, and it's because it's engulfed a bunch of these bacteria, and it's displaying it. This looks like a macrophage, and it's displaying on an MHC2, saying, I will help you. Let's go fight this bad guy. Okay. Now, this itself, the bacterium itself, is what's going to trigger a B cell. Remember, B cell receptors will bind directly to the pathogen, to the virus, to the bacterium. If it fits its B cell receptor, and if it is in agreement with the T cell, if it's a virgin cell, it will then divide into plasma cells and memory cells. All right. So what's the same about B and T cells? both B and T cells are specific, right? There's only one pathogen that they can make a clone army for. Their receptors can only recognize 1 to 2 antigens at best, 1 to 2 antigens at best. Therefore, we must have millions of virgin B cells and millions of virgin T cells so we are ready for any infection that might come our way, right? I don't know how old you are, but let's say you're 16, right? You're young. Okay, you got a lot of life left to live, and you're going to come in contact with all kinds of pathogens, and you need to have inside of you the B and T cells that can fight those future infections. Okay, so we have to have millions of varieties of virgin Bs and Ts prepared for any potential antigen in our lifetimes, All right, and then here, okay, little reminder, little review. Okay, here's a stem cell. Here's a T cell precursor. If it goes to the school of thymus, okay, then it can develop into a cytotoxic T cell, ultimately, which can kill anybody with an MHC1 displaying an antigen, or maybe it will develop into a helper T cell and bind to an antigen presenting cell, and they'll work together to fight off that infection. Now, T cells that don't stay in the school of thymus, those are your natural killer cells. Remember, we learned about them with our innate immune response, and they'll kill anything that has antibodies on it, and they will kill any foreign object, okay? Any foreign object. All right, but they just can't learn. So here you can see a target cell, and these are cytotoxic T cells killing this target cell. Maybe it's a cancerous cell or it's a cell that's infected. All right, so big picture here, big picture. Non-specific defenses, which we learned in video 1, they recognize cells or objects as foreign, they work to remove it. All of the non-specific defenses like inflammatory reaction, natural killer cells, protective proteins, they'll all work on a foreign object, okay? But specific defenses target the foreign cells object specifically for destruction. It's like they can only recognize one bad guy. Non-specific defenses recognize all the bad guys, but with that ability to recognize all of them, they're not incredibly good at their job. Whereas specific defenses take a little bit of time, like maybe 10 days to build up your clone army, but then they can get and wipe all of it out. Okay, so those are your B and T lymphocytes. They must be synthesized from just 1 to 2 virgin cells, so it can take about 10 days. They will both make memory cells, so you are faster on second exposure, so you won't end up getting sick. B lymphocytes make antibodies, specifically the plasma cells make antibodies that tag foreign cells for destruction, and T lymphocytes destroy our own cells that have been infected by those foreign cells or virus. So I made a little Venn diagram for you. So the target for B cells, virus, bacteria, toxin, they will make plasma cells that secrete antibodies that tag them for destruction. And here they will also make memory cells for next time. T cells target our own infected body cells. They will also make memory cells, but helper T cells to regulate and encourage the immune response, and cytotoxic cells for killing off our own body cells that have been overrun by that invader. All right. Again, you can see the stem cells, myeloid stem cells, red blood cells carry oxygen, platelets here. This myeloblast, notice it makes the granulocytes. The most prominent one is neutrophils, first on the scene, whereas lymphoid stem cells, this is where you get your B lymphocytes and your T lymphocytes and your natural killer cells who didn't end up going into the thymus. All right, so hopefully that's a good overview. And video 3, I will talk about these items: induced immunity, adverse responses, and disorders. And if you're one of my students, I will see you in class.